Hello, everyone. So this is session number 24. We are here in Nice. It's 9 a.m. in the morning, second day soon. We're going to talk about One Health. And so after 24 hours, it's really important to highlight that the ocean is actually offers a strong connection with human health. And it's exactly what we're going to talk about here with two focus maybe. One related to the potential risk when we pollute the ocean on our health. And the second one, as a source of medical innovation, when we protect the ocean, it offers ecosystem services like that. So this session explores One Health. So first on research on environmental epidemiology.
And then we're going to look at the development of bioactive compounds from marine organisms, such as coral-derived molecules, being investigated for cancer treatment. And then the broader message that healthy ecosystems are essential for preventing disease, supporting mental and physical health, and unlocking new therapeutic resources.
So in this penultimate session, we have the luxury of having two MARES alumni, Emma, Kelly Gonzalez, and Kelly Paul. Emma, you're now PhD students in Barcelona, and Kelly, you're PhD students in Marseille. Also joined by Rachid and Eric, who are more on the research side and startup. So Eric, you're a researcher at Université Côte d'Azur IRCAN, and Rachid, you're CEO of coral-derived, a startup based in Monaco. So first, we can start with you, Emma, if you agree.
Yeah, that's great.
So Emma, can you share first some information about who you are, maybe some of your background, and let's go then for your PhD on ecotoxicology.
Thank you, Christophe, for the quick presentation. So yeah, I'm an alumni from the MARES program. I've started my higher education in biotechnologies, actually, in an engineering school in biotechnologies. For the people that are not super sure about what are biotechnologies, I would say that they are, in a very simple form, just technologies made for living beings, but also made with living beings. So, for example, it could be technologies made for human health, so cancer treatment or anything like that for humans, but also they could be technologies made out of natural systems. So I focused a lot on water pollution and water system filtration during my studies. So it could be, for example, different plants or microorganisms put together as a natural solution for water treatment. So that's an example of biotechnologies. Then I realized during my studies that I was super interested into how to translate all this scientific knowledge about the world and how living beings work into society and more like maybe political systems in society. So for me, the MARES program was perfect in that sense, where we have this conservation aspect, very science-based, but also how it can translate correctly into societies. So yeah, I moved on with the MARES program, and from that, by the time I was really focused in this environment perspective, but then I discovered this whole field of One Health and environmental health, and for me it was perfect to kind of merge together these two worlds of humans and also nature, because it's not two divided and different things, but really from scientific knowledge, how can we share the message that we are all interconnected and how we don't work for humans or work for nature. It's like both at the same time, and just try to shift this perspective of we are humans at the center, like this very anthropocentric point of view into, and then nature, if we can, if we have time, if we have money. No, it's actually not like that. It's the whole thing together. So for me, it was super interesting to discover this field of studies, and that's how I started to do investigation and in that sense. So with a lab that is partner of MARES, I started to do my master thesis in this field, where we try to evaluate the contamination of methyl mercury in the Mediterranean Sea, and how this contamination is a burden for economies because it leads to disease and maybe poor well-being for every country that is, or for every person that is exposed to these contaminants. And so the objective was to bring these two things of both try to evaluate how much damage this contamination in the environment in the human health perspective, but also on an economical perspective, how much it costs.
Okay. So only connecting back all of that to also money, and maybe also to show that we have to invest money into nature if we want to maintain a good health for humans.
Yeah, exactly, and also maybe to show people that maybe prevention is better also in economical terms than treatments, because if we prevent disease to happen by reducing pollution, maybe it's more cost effective than just treating cancer later,
So seeing again investment in protection as a cost benefits analysis here. Is your PhD also into that now?
Yeah, exactly. So now I have shifted. I'm still working on the water element, but more like in cities, the water we drink instead of the Mediterranean Sea, and trying to estimate as well the exposure to microplastics and also the different compounds derived from that and the effect it has on health. And that field is called environmental epidemiology. So it's like how the diseases are distributed within a population and what are the determinants, environmental determinants of that disease. In my case, chemical exposures. Yeah.
Okay. Thank you, Emma. Maybe you want to share already one of your first results, something that is more impactful for you? We can come back to that after that during the question.
Yeah, microplastics is a very tricky field because it's been very few years that we can actually study microplastics in the human bodies. So right now, the research is at the very early stage of like trying to develop methodologies to measure them properly and so on. But so we are. I couldn't have any certainty on the results for now, but what's for sure is that it's everywhere and it's super hard to study because it's just everywhere. It's like in the air we breathe. It has been detected in the clouds, in very high mountains, but also in the deepest oceans, in our food, in the human body as well, so in different organs and tissues. So we are just starting to evaluate the
extent of the issue. Yeah,
exactly.
Thank you, Emma. We're going back to that in a few minutes, and we're jumping to Kelly. So before Kelly explains a little more in detail her work in Marseille, Kelly, I propose we have a look to your 1-minute video.
Hello, my name is Callie Paul, and I am a second-year PhD student with the Mediterranean Institute of Biodiversity and Marine and Continental Ecology in Marseille.
My project exists under the ANR Le Coco, Living with Chlordacone and Opportunity-Based Co-Construction. Chlordacone is an endocrine-disrupting insecticide that was widely used in Martinique and Guadeloupe despite its known health risks. Although it was banned over 30 years ago, it continues to heavily pollute both islands today. My part of the Le Coco project is to elucidate the effects of chlordacone cocktail pesticide exposure using the sea urchin, Paracentrotus lividus, as an experimental model. The goal of my project is to provide data to environmental lawyers to support new legislation on cocktail pesticides. Ultimately, the project aims to contribute to remediation strategies for both people and ecosystems in Martinique and Guadeloupe who have been disproportionately exposed to chlordacone due to the long-term impacts of colonialism.
So Kelly, your study here is part of a general context, which is really important, really famous in France. It seems to be an end-to-end project, really from studies to action. What exactly are you doing, Kelly, in this project?
So thank you for the introduction. The main goal of my project, which is just a small portion of the ANR Le Coco, the ANR Le Coco exists under federal project called Plan Corticone, which has different sections. And so this is actually one of the 5 plans. And my specific part of the project is to better understand how corticone cocktail pesticides are affecting gene expression. And so we're using the sea urchin, which has approximately 70% of the same genes as humans, as an experimental model. And the idea is that while we have studies of corticone as a single molecule, and we have studies on vertebrates as well with corticone exposure, and so we know that it's carcinogenic, we know that it's causing cognitive problems, developmental issues. The problem is much like Emma was stating with plastics, we have pesticides everywhere, and it's never just one single molecule or one single pesticide. So in Martinique and Guadeloupe, well, we have a huge problem with corticone contamination as it does not degrade. It's classified as a persistent organic pollutant. We also have roughly 63 molecules that are also found in water samples alone. So the problem is that when we have a cocktail of chemicals or a cocktail of pesticides, it changes the biological response. And what we found with corticone is that in toxicology, we typically have a standard dose response model, and it's the higher the dose, the higher the biological effect. The problem with corticone is that it has a non-monotonic dose response, meaning that at a very, very low concentration, it can actually have a very high biological response. And so the issue is that when it's mixed with other pesticides, as it exists in the environment, it can have a very, very, very high biological response with a very, very low dose. And that's the problem, as we have different potability levels for water, and we have different legal levels for how much pesticides we can have in our foods, in our water, not just pesticides, but other molecules, other chemicals as well. And there's a great issue with that, is because when they're mixed, the biological response and the effect could change.
Yeah, so it's crucial not to study the effect of each of the contaminants one by one, even if I guess it's also important in science to adjust one variable at a time, but also to put everything together. Maybe we can also talk about that with Eric. So Kelly, how are you doing that on a day-to-day basis in the lab?
So I actually conduct experiments with about 120 sea urchins. We have 18 different conditions, including one control. And while I said we found 63 molecules in freshwater, below dew, in Martinique, there was a study that went on for about 5 years. And there was auto samplings of fresh water from a river that were taken weekly. So we have these weekly composite samples for about 5 years. And what we found is 63 molecules. So we can have an idea, 63 chemicals that are in the water simultaneously. So it's very difficult to, from a methodological point of view, to conduct an experiment with 63 different
63
chemicals.
variables.
Because I need to
How long is your
construct-
PhD? 10 or 15 years?
It's never ending. I'm going to be a student forever. No. But so the idea was that we were going to start with the pesticides that are most abundant. So we actually started with corticone because it's really found
Yeah,
everywhere.
we know also the controversy, especially with this molecule.
Yes, and it's unfortunately a huge social problem. Imagine, we're in France and in Martinique and Guadeloupe, you're drinking bottled water because the tap water is contaminated. So anyway, we started with corticone, and these are all at environmental concentrations, and then a aminomethylphosphonic acid, which is the primary degradate of glyphosate. And glyphosate is actually in France and was just approved from the EU to be used for 10 more years. And glyphosate also has connections to carcinogenic effects. And then we also are going to be, well, we conducted this experiment with azoxystrobin, which is a globally used fungicide. So what we did is from these water composite samples, we selected the 3 based on the prevalence in Martinique and Guadeloupe, but also from a global perspective. And with my 120 sea urchins, we had 17 different concentrations, low dose, but at environmentally relevant concentrations. And they were exposed to these cocktail pesticides for 2 weeks. What we did afterwards was, unfortunately, I dissected the sea urchins and took their DNA from the gonads, which is actually, if you eat sea urchins, is what we eat from the sea urchins. So I took this sample, and they're now being processed to be sequenced in a traditional RNA-seq analysis.
So with the RNA-seq analysis, the idea is to see what part of the DNA is more or less expressed due to the exposure to the contaminants.
That's the idea. And so we really want to see the patterns of gene expression in different concentrations with key genes that are associated with endocrine response, as it is an endocrine. It's a proestrogenic chemical. And typically in endocrine disorders, we're having a multitude of health effects in humans. And while my project is a very human-based, it's very human-focused, as unfortunately we have humans that are being poisoned with chlordecone. There are also very strong links to chlordecone contamination and lack of biodiversity in the oceans surrounding Martinique and Guadeloupe as well.
Thank you, Kelly. I think there is a good transition also to Eric. So Eric, you're a researcher at IRCAN, so it's in Université Côte d'Azur, and you're also using different biological models related to aging, cancer. So can you tell us a bit more about what you're doing in this context?
Exactly. So first, thanks for the invitation to join this very interesting panel and very complex panel about One Health. So just a brief background. So I'm a molecular biologist, and I'm specialized in cnidarians, jellyfish. Well, we're not working on jellyfish, but jellyfish, sea anemones, and corals, because they have a very interesting biological and physiological capacities, which are able to respond to stresses that would kill our cells. They are able to regenerate when we cut them in different pieces, and certain of those cnidarians have an extended lifespan. And so in the Institute for Research on Cancer and Aging, we're interested in trying to understand how those animals are able to fight stresses, how they're able to regenerate, and how this molecular underlying process is linked to the longevity of those animals. So we really try to focus on the genes, and then potentially in the long term also translating this to mammalian models to see what can be sort of transferred and translated into therapies. But this is really fundamental research. And similar to what Kali said about the sea urchin, what is really interesting, even though that cnidarians, corals, sea anemones look unlike humans, they also share about 70-80% of the gene content. So what we find in cnidarians is mostly related to what we can also find in humans. So there's an interesting connection and link to potential human innovations.
That's the idea of biological models which are used. It's because these models, or these organisms, even if they look really different to humans, they share a big part of their DNA with us, so we can study them.
Exactly. So most of the molecules that we identify have similarities with human or mammalian orthodox. Exactly. And what we have not investigated yet, but which might be really interesting, is because of the high level of similarities, and so there are only a few genes that are sort of specific to variants, the chances of those genes to interact with homologs of human genes is very high. So a gene is never working alone. It's always a complex of proteins. So this is something that I think is really interesting to look more into depth, how they interact. And this could be linked to bioactive molecules to treat cancer, to treat other diseases, or to have new targets to fight against certain diseases. So this is what we do in the lab, and I just wanted to come back also to some stuff that we do in the field, because I think the One Health concept, like you introduced, is really on both sides, and I think it's a really complex aspect. So there's one side that is trying to find new innovation that could help human health, but at the same time, it's also to find approaches to determine how the marine environment is sort of affected by the human impact and how this affects locally. So we also have projects to look at the stress response or the health of the coral reefs, which are sources to food to a lot of people around the world, and using non-invasive approaches, combining imaging and AI approaches. And I think you had a session on AI yesterday in this around the world. And I think this is really what fascinates me in this line of research that we do, is that you can also combine fieldwork, where we have really close interaction with the communities, and also work in the lab, where we can try to go further and dig deeper into the molecular mechanisms. And this is probably also the transition to Rashid that is then pushing the next level to really human application. So this is not a coral, but it's an octopus, but it's just to illustrate the term that I think is really fascinating, the bioinspiration, because we can learn a lot from those marine organisms. And octopus, they inspire a lot of robotics for microsurgery, for example. And I think then the last photo is a clownfish, which is cute and might be completely unrelated, but there are also they are able to live in sea anemones and not without being stung by the tentacles of the sea anemone. And so there's some interest in trying to understand what does the mucus contain to protect them, to get sort of new approaches to put on the skin so we don't get stung by jellyfish. So I think once we spend more time on the organisms, you can learn a lot, and I think it's very inspiring.
Thank you, Eric. And it's also an excellent transition to Rashid and the work of Rashid at Coral-derived in Monaco. So we talked first with Emma and Kelly about the fact that our impact on the ocean can have negative consequences on our health. We start to understand that. But on the other side also, Rashid, when the environment is in good health, the biodiversity on a place can be useful to create treatments. And for example, here at Coral-derived, you started with treatment against cancer. Can you tell us a bit more about you, Rashid, your background, and also the beginnings first of Coral-derived?
Thank you, Christophe, for the invitation. And yes, first I'm a genetic researcher. I work for more than 15 years in therapeutical strategies for genetic disease, for human genetic disease, in Paris and Versailles, and in private companies too. And in 2015, I arrived in Monaco to work on these topics first, but we started a scientific collaboration with the marine biologist that had a technological problem to produce molecules from the corals at large scale with a very high purity, just for fundamental investigations. And for the human parts, I had this technology on my hands, and I decided to start this collaboration. And we produce a really large quantity of the first molecule from the red coral of Mediterranean. And we discovered very interesting innovative properties of anti-UV protection and anti-cytotoxic protection against the skin. And we decided to patent these innovative properties with the Scientific Center of Monaco, which is the academic research center of the Principality. And immediately we had some big groups of cosmetics that came to speak with us about the possibility of the valorization of the coral resources. And the researchers themselves show me a list of genetic sequences coming from these coral organisms. And we realized the huge potential that exists inside these specific organisms. And we discussed with the Monegasque authorities about the possibility to have a virtuous circle inside the Principality between the fundamental research and its application. And that's why we decided in 2018 to create Coral-derived in order to go through the valorization of the coral resources. So Coraliotech now is a marine biotech company whose the main mission is to go through the valorization of these coral resources by identification, production, and commercialization of bioactive products for human health and well-being. To do that, we use a sustainable approach that combines artificial intelligence, genetics, and biotechnology. And specifically, we use the DNA of the coral organisms as a raw material to produce in the laboratory products that are 100% identical to the natural molecules, with, as you can understand, zero environmental impact. So this is a very important part of our activity. So today we have a genetic database containing nearly 12,000 exploitable sequences, from which we select the most scientifically and applicably promising ones for different sectors of activity. The most important one is the health, and we are currently developing molecules aimed at applications in cancer, infectious disease, inflammation, dermatology, and many other areas of human health. In fact, the quantity of application is very huge, and we are working now to try to find the best ones in terms of industrial transition, because you have to understand that one of our challenge is not only to produce in R&D scale the molecules and to prove their efficiency, but also to implement the industrial production in order to respond to the scalability of the industrial clients.
It's fantastic what you're saying, because I think it's important for everyone to understand that when we say that we find a nice molecule, a nice sequence of molecule from coral reefs, the idea is not to collect the corals and break them in pieces and produce the treatments against cancer with that. You synthesizing the molecule. But how long does it take? And the first time maybe it was longer, but now with your technique, how long does it take to say, oh, there is a sequence that creates this molecule? With your technique, can you recreate any molecule, or you have some limitations, and how long does it take?
Yes, there is different questions in your one question.
Yes, often.
First, it depends of the sector, of the targeting sector. For the cosmetic ones, it's more easier, and it takes 1 year between the identification of the sequence and the first commercialization of the product itself. We have 4 different steps. The first one is the manufacturing process. Try to have the better yields and high purity. This is 2 of our objectives in terms of specifications. The other step is the toxicological evaluations of our molecules. And the third and the most important for me step is the functional evaluation of the molecules. We perform different kind of tests, assay in vitro, ex vivo, and for pharmaceutical approaches, we need to go through in vivo evaluations and clinical trials. For the cosmetic ones, we are already passed through these 4 different steps until the end of the clinical test of the molecules. If we found innovative properties, we decide to patent these innovations in order to sell licensing to the future clients. But yes, 1 year is for the well-being sectors, and probably 2 to 3 years for pharmaceutical.
The pharmaceutical, of course, yeah. Yes. Much more hurdles, I guess.
So, I mean, I have plenty of questions, but we're running out of time. So I was just wondering, before we go, if each speaker could give us a kind of like a 30-second takeaway message about the importance of the One Health kind of like framework, or maybe of its key challenge, or maybe its key promise or hope.
Yeah. Start with
maybe with
first one
with Emma.
Allez, Emma Kelly.
It's a hard question. I think what's the key message of this session is maybe it was super useful for me to see all the different perspectives and just see that it's we are dealing with very complex systems, so that are all super connected to both human health, nature, economic systems, political systems. So for me, what's make a really good scientist or other professional would be to really encourage students and people to have these very complex system thinking skills, because it's what makes science useful at the end, to be able to implement it in the real world, but also do useful science from the beginning of the thing. So for me, it's really what I want to emphasize.
Open your horizon. Thank you, Emma. Kelly.
Thank you. I would also have to agree with Emma that it's very important to have a robust style of thinking to understand that when we change something in the environment, specifically humans, we have a butterfly effect throughout many different systems. I think the key takeaway of the One Health in the perspective of my project specifically is that lawmakers and policymakers and governments need to be relying on researchers and these broad, robust perspectives in order to prevent these butterfly effects, these multifaceted contaminations that are ultimately leading to One Health. So One Health is great for the environment and for humans, but it's also a great perspective moving forward about making policy decisions that better support not only our environment, but human health as well.
Yeah, pushing the concept to the end. Eric?
Yes, I'm in line with that. I think the One Health concept, as I mentioned earlier, is quite complex. What I think is really important here is to consider not only the effect of pollutions on the marine environment and directly the human health, but also on the impact on the organisms that, for me, are really a source of innovation, of hope to cure disease. And of course, I think it's important to, on the one side, protect the environment just because it's part of our daily life and it's important for our survival. But in addition to that, I think there's really a source of hope for the future. And I think what Rashid does is really in line with that. And we should preserve this for so many reasons. And I think that would be my main message, that in the One Health global thinking, there's so much important aspect and everything is connected. So we can't disconnect it. And I think it's really the term of One Health, and we're going from protecting the environment to saving our lives through innovation. So I think it's great.
Rashid, no pressure, but you have the final words of the final presentation based on the scientific topic.
No, I don't have pressure, but Eric resumed very well what I wanted to say. So there's nothing else to say, just to confirm that we need to preserve this marine environment in order to have the possibility to find some resources able to go forward to save lives, to save human lives, because we know that the ocean has a large potential to do that for the future generations.
Thank you, Rashid. It's an excellent transition also to the next and last session, session 25. It's going to be about scientific diplomacy and the roadmap of UNOC with University Côte d'Azur, and we're going to talk about BBNG. So the aspect of protecting the deep sea before we destroy it. So yeah, thank you to all of you.
Thank you very much.
Thank you on the scientific topic. And I hope you will stay with us for the last one coming up in 30 minutes.
Last dance?
Yeah, the next one we have the vice president, so we cannot do that. Bye-bye.
Bye. Thank you.